When you heat a DNA sample up to 95 degrees, the double helix separates into two raw strands. For an enzyme to bind accurately in this environment, it requires a primer sequence that meets specific thermodynamic conditions to outcompete billions of other potential binding sites. To create that binding site, you have to build the sequence yourself. We are going to design, mathematically balance, and engineer a set of PCR primers from scratch. To follow along, you will need your target gene sequence oriented 5' to 3' and a basic calculator. Replacing guesswork with a strict thermodynamic checklist reduces the risk of non-specific amplification and increases the probability of achieving a high-yield reaction. This 5-point checklist defines the anchor, the specific region of the primer that binds to your target DNA. First is length. You want 18 to 24 nucleotides. This range provides enough specificity to find your gene without being so long that the primer folds back on itself. Second is the melting temperature, or TM, which should fall between 50 and 65 degrees Celsius. We calculate this by multiplying the number of A and T bases by 2 and the G and C bases by 4. Rule 3 focuses on parity. Your forward and reverse primers need melting temperatures within 5 degrees of each other, so they attach to the template simultaneously during the PCR cycle. Fourth, aim for a GC content between 40 and 60%. This supports the fifth rule, the GC clamp. To ensure the polymerase can start copying effectively, the final 5 bases of the 3' end should contain 1 to 3 G or C nucleotides. These 5 metrics provide the physical boundaries needed to guide a successful amplification and prevent the reaction from failing due to structural issues. We'll start with this sample target gene. The top strand, reading 5' to 3', serves as our initial template. The forward primer sequence is identical to the first 20 bases of this top strand, as it is designed to bind to the complementary bottom strand. Let's verify the anchor. At 20 bases long, it meets our length requirement. Counting 10 Gs and Cs out of 20 total bases gives us exactly 50% GC content. Using our formula, 10 AT pairs and 10 GC pairs, we calculate a melting temperature of 60 degrees Celsius. The sequence contains no long repeats that might cause slipping. Finally, we look at the last 5 bases, ACCAA. It contains a single C, which satisfies the GC clamp rule. Because this sequence passes every check, we can be confident it will bind securely to the template without forming unintended structures. Now we look at the end of the gene. To bind to this top strand, the reverse primer requires a different approach than a simple copy. We generate this sequence using a 2-step process called the reverse complement. Step 1 is the complement. We swap every A for a T and every G for a C along the final 20 bases of the gene. Step 2 is the reverse. To ensure the sequence reads in the standard 5' to 3' direction, we flip the entire string backwards. We then check this against our rules. This reverse sequence also has 10 Gs and Cs, giving us a matching 60-degree melting temperature. Matching the temperatures of the forward and reverse primers is essential. It ensures both sides of the gene are primed for copying at the same moment in the thermal cycler. While these anchors will amplify the DNA, you need additional sequences if you plan to insert that DNA into a circular plasmid. An engineered cloning primer uses a 3-part architecture: a spacer, a restriction site, and your target anchor. We start by placing our validated forward anchor into the final segment of the design. Next, we add the recognition site for your chosen enzyme, such as GAA-TTC for EcoRI, directly to the 5' end of the anchor. Finally, we add a short spacer, like CGAT, to the very beginning. This provides the physical DNA padding the enzyme needs to sit on while it makes the cut. These engineered overhangs provide the PCR product with the specific molecular architecture needed to integrate into a destination plasmid. You now have 2 fully engineered 5' to 3' sequences, mathematically balanced and ready for synthesis. These final strings are what you provide to a commercial manufacturer. Most ordering platforms, like the one shown here, require you to input the sequences in the 5' to 3' orientation. Before finalizing your order, it is best practice to run your design sequences through an automated primer calculator to confirm your manual calculations. While laboratory variables always exist, adhering to these thermodynamic rules removes the most common causes of primer-related reaction failure and provides a reliable foundation for your cloning project.